webinar. It's on the application of RNA interference to manage fungal diseases. And I think it's a very important topic for RNA interference nowadays, not only because fungal diseases, as we know, cause the most severe losses, economic losses in crop production all over the world, but also because of the forthcoming restriction in pesticide usage, at least for Europeans, that they are planned, they are announced in the Green Deal of the European Commission. This restriction will probably give quite some trouble to the farmers because they are now managing a lot of these fungal diseases with pesticide that with active ingredient that will be banned very soon, and they do not have any real effective alternative. RNA interference has been, I mean, for the number of publications, the scientific report that we see every day is a very promising alternative. We know about the regulatory issues that will take some time, but for sure can represent a good candidate for developing some solution. So I don't want to steal more time to the speakers, which I thank a lot, all of them. We have very nice set of speakers coming from different parts of the world, like Canada, United States, Brazil, Germany, and also Italy. And so for me, it's just a pleasure. I don't want to steal more time. And I introduce the first speaker, who is Professor Kogel from Gießen University. And he is a professor at the Institute of Phytopathology. And he is working in plant microbe interaction since a lot of time. He got his PhD in biology in Aachen University in Germany. And he's professor at Gießen University since 96. He will talk today about the disease control by double-strand RNA. And yeah, I just please, I just welcome you, Carl. Just go on if you want to share your screen with the presentation. Last thing, as Bruno already said, I ask the audience to write the questions on the chat, and then I will, at the end of each talk, I can read, and then we will have a final discussion at the end of the webinar. Thank you, Carl, please.
Yeah, thank you very much, Elena. I will share my my screen now and hope it works. Okay, you see it. Good afternoon from Germany. I am currently here, as you see, in the middle of Germany at the University of Gießen, which is close to Frankfurt. What I want to say before I start here, we uploaded the talk and all the data I will present today on open access in our university server that is approachable by all people. So it's public already. Yeah? So it's more or less published. I start off here with the situation Elena told us already. There is a lot of problems concerning the use of active substances, especially probably in EU. because we have a cut down of more than 50% of pesticide types, and more important, if you look at the situation in cereals, 9 of 10 of the best-selling fungicides will be out in the future, in the near future. So we will have, or farmers and consumers will have a lot of problems. Our solution is, it is just one solution, of course, not the only solution, using non-coding RNA. And I start off here with a picture which shows you basically the discovery of helingin in 2013, and it was published also with animals and animal pathogens. That RNA is exchanged by organism and function as signal molecules, and we call this cross-kingdom RNAi. So I don't go in details, but from the application point of view, it's interesting that we can learn from this natural situation, and we could use RNA and especially a lot of different types of RNAs for disease control. And we have proof of concept, of course, for some situations here. For instance, the control of viruses, there is a clear proof of concepts even in fields. And also we have products on the market to control insects. But all the other pathogens and pests, yeah, the situation is different. We have not enough information to tell whether this is effective or not. So I give you just 2 slides that shows how we can learn from insects, because we did a lot of work in insects. So insect feeding is very easy. With double-stranded RNA is very easy. As you see here, we used artificial diet with double-stranded RNA, and this double-stranded RNA, which has a fluorescence marker, accumulates in the insect guts. I don't go in details, but you should realize that the efficiency of the uptake is very different depending on the experimental design. You have for Higgs very efficient activity of double-stranded RNAs in some insect groups. That also differs a little bit, but we don't go in details. We have very efficient gene silencing when we offer the double-stranded RNA with the artificial diet. But if we use 60, at least if when we worked with aphids, is very low. The efficacy is very low. So the last slide for insects. What can we really learn? This is a complicated slide, and it is published. You can go in the source here, Advanced Biotechnology 2020. But what I want to show is here there are probably 2 main possibilities that RNA is transported from the gut to cells. And that are receptors that are not found in fungi, these CYT1 and 2, RNA receptors and transporters. There are RNA binding proteins that are probably used, and there is the vesicle transport or transfer based on clathrin. So these 2 last possibilities might also be found in fungi, and there is a recent report that Clathrin-mediated transport of RNA plays a role in fungi. So it's very important to realize that insects, the efficacy of or efficiency of RNA uptake is dependent on other factors, like the length of RNA. In some insect groups, there are chemical formulations already published that support the transfer, and the RNA stability is very much affected by nucleases in insects. So what is the situation in fungi? We had, in recent time, we defined a test set of fungi, mostly fungi that go to cereals, but not only. This is the test set here. Rhynchosporium zecalis is a very important pathogen on barley, but you know verticillium, but this species here is very narrow in host range on Brassicaceae. Then we have Piriformospora indica, which is a endophyte, a beneficial fungus, and goes to root, not to leaves. And we have Zymoseptoria tricity that is already investigated a lot. And if you go here to the last column, you see the situation with RNA uptake. It is not published for Rhynchosporium. It is published for verticillium. It is not published for Piriformospora, and it is published as not functioning or not working in Zymoseptoria already. So we go in details with this set of fungi here in the talk. So I start with Rhynchosporium commune, which is my favorite fungus. Is a very nice fungus, and you see the very, very impressive disease phenotypes here of the fungus on barley. These lesions here. The fungus is hemibiotic. It grows under the epidermis, under the cuticle, and then penetrates to form lesions. And the conidia are very typical. We used this well plates approach to test the uptake of double-stranded fluorescence double-stranded RNA, and we start with 420 nucleotide long RNAs. What you see, and it's not very astonishing, there is a clear uptake by Rhynchosporium. And I have to say, we started these cultures with conidia, and then immediately we added the double-stranded fluorescence double-stranded RNA. And what you see is, I don't go in details, there is a clear uptake in Rhynchosporium for this RNA. This is fluorescent RNA to 420 nucleotides. Is there a correlation with silencing? We used here another RNA. This is a 481 nucleotide double-stranded RNA directed against the GFP gene sequence, and we used a reporter fungus that produces GFP. And what you see here is a clear time-dependent suppression of the GFP expression when we fed this double-stranded RNA, indicating there is a silencing after transfer of double-stranded RNA from liquid cultures to the fungal mycelium. The same is true for Verticillium longisporium. As you see here, don't go in details. There is also uptake when we started the cultures from conidia. Again, the same double-stranded RNA here, fluorescence, and 420. So again, a correlation between uptake and gene suppression or suppression of gene activity. We used in all probably 5 here. I show 4 target genes that are really downregulated after feeding of specific double-stranded RNA against these target genes. I don't go in details here, but you see it's very consistent that in all situations, these target genes are downregulated by the 420 nucleotide. We were interested to see whether we have a bias here or a preference for length of double-stranded RNA in fungi. Why? Because we have this bias in insects. So it's very important to see whether the bias is also here in fungi. We start with verticillium again, and you see here we fed 21, 402, 1,451, and in all cases, this is just an overview here, you see a accumulation of fluorescence in the mycelia, indicating we don't see a preference. Now I go to another fungus, which is interesting because it's an endophytic beneficial fungus and it goes to roots. And you see here a barley root stained with WGA alexaflor, which is specific for chitin, therefore the green color. This fungus induces growth promotion, which growth promotion and induced resistance. And as you see here, even under field or near field test conditions, you have a growth promotion. As you see here, non-treated and treated, and there is a difference. And of course, the statistics is also there. So indicating the fungus is really beneficial. So we fed spores again with double-stranded RNA. In this case, it is 21. And as you see here, after 1 day already, you have uptake of fluorescence from this double-stranded RNA, and that accumulates later on after 2 days. This same is true with the 420 nucleotide RNA. As you see here, there is accumulation in spores and there is accumulation in mycelia, and you have it probably most concentrated in tips of the growing hyphae. And what you also see, but this is still speculation, that the spores that did not germinate have still fluorescence, and upon germination, of course, all the material goes from spores to hyphae, and then you don't see anymore the spores, indicating that they are really inside, the double-stranded RNAs are inside. As this is just another picture here with the 420 nucleotide RNA, and you see very, very, very sensitive uptake of double-stranded RNA with this beneficial fungus that goes to roots. And we could also we could not detect any preference again for length of double-stranded RNA. This is 700, this is 1400, and even here, even longer. You see there is still uptake of the RNA. We don't know yet whether the efficiency is the same, but we don't see differences in fluorescence. But of course, there we need more experiments to demonstrate whether there is a silencing efficiency dependent on the length of the RNA. But in principle, you see there is no preference in fungi. So I want to show you just a few slides with alternative screening designs, because we are not sure in the moment whether using the liquid cultures is enough to tell people these fungi are really sensitive to gene silencing. We have some indications that we need the typical 6 experiment to finally demonstrate really that the double-stranded RNA works and the fungi, a given fungus is amenable to it. So alternative screening designs, we have here used Arabidopsis in this case. Arabidopsis usually is not very sensitive to 6, but what we did here, we fed the RNA 21 nucleotide fluorescence RNA via the petiole. And then we used a Arabidopsis reporter line specific for the phloem, as you see here, using the SAC2 reporter promoter and YFP reporter, and then we incubated with CYP3 RNA, and what you see is in the merge picture there is no overlap in the colors, indicating that this goes really as was expected via the xylem, and there is no, as far as we see, no exchange of RNAs inside the plant from xylem to phloem. A last experiment I wanted to show you here is the treatment of Arabidopsis leaf with fluorescence double-stranded RNA, again the 21 nucleotide, and then again working with the Arabidopsis reporter line with the SAC2 promoter. And what you clearly see here is, of course, a phloem, and then the sub-3 RNA fluorescence in the xylem, and this is the merge here. There's no connection and no overlap, indicating that if you treat leaves, it goes more or less, at least in Arabidopsis, to the xylem. Okay, what can we learn here from these experiments so far? Pests, exogenous double-stranded RNA is a promising and very efficient tool. We know that already. That will come and is already in the market, and there's no doubt that it's efficient and it's a very good tool in the future. Fungi, we need more information. We don't know. I'm not sure whether we can develop it to the market, or and whether farmers will accept it and the consumers will accept it. I'm not sure. There are still open questions, but you see in some systems it may very work very nicely. What is needed probably, that is one of the priorities, in innovative compositions of formulations to enhance the uptake of double-stranded RNA to fungi, in fungi. What we also learned from the experiments I have shown, that double-stranded RNA can move inside plants, even if you treat the leaves, that is clear, but it goes more or less the xylem and the apoplastic path probably. And this is it goes to probably the xylomatic root or the apoplastic path. So this is the end of the talk. I hope I kept the time. There are many people working here in this field now in our department. Jafar Imani and Maria Ladera Carmona, is a new postdoc focusing on these aspects here. And Asesik is a postdoc from my lab that made a lot of experiments, research on RNA interference genes in cereals. And there are many other people, and this is very much supported, the work, or was supported by industry, but now it's most of the work is mostly supported by public money from the German Research Council. Thank you very much.
Thanks, Kalle. Thank you very much. Very nice talk and very nice work on fluorescence. There are two questions. I'm going to read it. Actually, three questions. So one is, have you analyzed Dsr-mediated processing of the applied double-strand RNA in fungi? This is the first question.
I didn't get the question. What is with Dsr? I understood Dsr.
Yeah, it's asked. The question is, have you analyzed Dsr-mediated processing of the applied double-strand RNA in fungi?
Yeah, we have done a lot of work with Dsrs in different fungi. The first information is Dsrs are very sensitive, or let's say silencing Dsrs or knockout Dsrs reduces the virulence in general very much in many systems. including fungi that don't have them shown here, like Matapothe, very, very strong phenotypes if you silence dicers. And we have also done some silencing with double-stranded RNA in addition to knockouts. And here you find also an effect on at least on 6. We did not work on liquid cultures here, but we have published already that knockouts of Dicer 1 and 2 in Fusarium affects the 6 strategy to control the fungus. So yes, there are a lot of information on dicers.
Carlo, it's Michael Wassenegger asking this question. I want to know whether you checked whether the uptake, the double-stranded RNA that was uptaken, the fluorescent double-stranded RNA, was processed into siRNAs by the fungi dicers, because you have shown the uptake but not further processing.
No, we have shown that the silencing works, yeah. So I showed you experiments. These are very important in 2 fungi that the double-stranded RNA is taken up and there is a silencing of the target genes. Yes, but we did not follow other experiments here where we investigated exactly the dicer products by isolating the degradation product so far. But silencing is affected.
Okay, let's go on. There is another couple of questions from Francis Fang, who is asking, to me, the uptake seems more on the surface of a fungal Ife. Did you think about using dual dyes? For example, colocalization with FM464. And the second question he has is, how did you detect silencing of the targets in the in vitro experiments? Did you have any data on the lifetime of silencing in vitro?
Yes, I showed one experiment where we have the silencing for 6 days. You know, the silencing of the GFP reporter gene could be demonstrated for 6 days at least, and there was no release of silencing, so it is longer in the liquid cultures. But these experiments are maybe not so interesting in liquid cultures because okay, this is a quite artificial system. But yes, there are information on that. And then, yeah, it is always the question where the RNA really is. And Helen Jin will tell probably more about that. You should have a control for the sRNA degrading enzyme with the double-stranded RNA degrading enzymes. That is one point. The other point is we demonstrate silencing, so it must go in. Yeah, so that is logic. And if you do the confocal laser microscopy and you have some experience in that, you see it is inside.
Okay. Then there are another couple of questions. I think we still have some minutes. Yeah. Okay. One is from Novio Concepcion. I am sorry, but I have. Oh no, sorry. This is not a question. So another question is: have you ever faced with the side effect of non-target double-strand RNA to fungi?
Okay. Can you repeat it again? Sorry. Have you?
Have you ever faced with the side effect of non-target double-strand RNA to fungi? Maybe it's a spelling mistake. Have you ever faced?
Yeah, there are old publications from our group in 2016. We have demonstrated or we worked with the Site 51 cytochrome 51 target genes, and there we had the issue of side effects. So I refer to this Koch et al. Publication 2016 in PLOS pathogens.
Okay. Yeah, maybe there are also this one to better address double-strand RNA length and other conditions. Did you try simultaneous uptake of different double-strand RNA tagged with different fluorescence color? But this was, I think you already answered to this, no?
Yeah, but we did not do that.
Okay, thank you, Kalle. Then at the end of the webinar, we will have more time for discussing. So let's go on now. Yeah, next speaker is Professor Gulin Eduardo from Brazil. He is associate professor at the Federal Institute of Education, Science, Technology in Santa Caterina in Brazil. He's obtained his PhD in Brazil at Unicamp, is a state university of Campinas. And he's done a lot of work since quite some time in the interaction between Colletotricum and citrus species. So he's today talking about RNA interference in Colletotricum abscissum, causal agent of citrus PFD. Please, Eduardo.
Hi, hello. Just a minute because I have to. Here. Yeah. Can you see it?
Yes.
Yeah. It's okay. I'm present. Yeah. Full
Very
screen.
well. Sure.
Okay. Okay. I was muted because I forgot to switch it to unmuted. So. Hello, everybody. So I would like to start this, say thank you to the webinar organizer, especially Professor Elena Beraldi, who invited me to give this short talk. I will more specifically talking about Colletotricum species that cause disease in citrus, so quite different than Professor Kugler before, who spoke a lot of different kind of fungi, and I will be quite specific to Colletotricum. Yeah. And of course, the RNAi interaction and a way to control this fungus on the field, maybe in the future. So maybe, as we may know, the citrus tree is a kind of tree that stayed for a long time in the field, and sometimes for several years or 10 years, it requires some kind of special condition, for example, soil, plant, and select the correct variety. The breeding is quite important for that. And during this all time in the field, they are more open or susceptible to be affected by different kind of pathogens. So such as bacteria, virus, and of course, fungi, different kind of fungi species. One of these fungus species cause this specific disease called post-bloom fruit drop. So this disease is quite important and very frequent during the rainfalls. So here in Brazil, we have a large production of citrus. As we may know, we are the largest producer of citrus juice in the world. And this specific crop is really important. So when the fungus are present on the crop, we can see this kind of brownish lesions on the citrus petals, and these lesions, together with the weather conditions, high humidity, for example, can spread these spores all over the citrus flowers and infect all the citrus flowers in this specific tree and the close trees also. So then this fungus can affect the young fruits here and induce the premature fruit drop, and it causes a large economic effects. So therefore, it's so important for us this specifically disease. But sometimes we start to think about which kind of fungus or fungi can cause some disease, and here we have some problems at the beginning. Maybe most of you knows about Sclerotinia acutatum species, but in 2011, sorry, Uric Dam have divided this Sclerotinia species this Sclerotinia species to a species complex of Sclerotinia species, and one of these new species is called Sclerotinia abscissum. Therefore, I'm talking today about Sclerotinia abscissum and not about Sclerotinia acutatum. Maybe some relation about this uptake and RNAi inducing can be similar, but we are not sure about that because they are different species. And so I isolated this one fungus from the field, and after phylogenetic studies, I realized that my isolated belongs to a Sclerotinia abscissum species that was published by Pinho and Pereira in 2015. Yeah, and belongs to the Sclerotinia acutatum species complex. Just to show you how the Sclerotinia abscissum looks like, and we can see here the macro morphology and the macro and micro morphology of this Sclerotinia abscissum in the plates and microscopy here. And similar to different most of the plant disease, the control of this post-bloom fruit drop or for the control is using chemical compounds. So it's quite useful or quite often the producer are using chemical controllers to different kind of pathogens and disease, of course. So, but we have many classes of different chemical fungicides. So among them, we have those that cause affect cell respiration, and it's used to control many of Sclerotinia species. So in the field, for example, the application is variable from 7 to each 14 days. So it means a lot of money spent and environment damages, and a lot of things are related with this chemical control. Moreover, we have resistant reports. So we have to think, as we are doing today here, and everybody used to do at their labs, we have to think about some alternative strategy to control this disease and another different kinds of disease, of course. So one of these different alternatives is the RNAi that we are discussing today here. And the RNAi are a promise strategy to alternative control. And there is some reports about this RNAi applied to Sclerotinia species, but not to Sclerotinia abscissum. So when we think about RNAi, I mean, when we start to work with a different pathogen, or it's not well known, the process is not well known as some models pathogens, we have to think about some questions. So if these RNAi are functional or would be a functional strategy to Sclerotinia abscissum control. And another question can come, for example, if the Sclerotinia abscissum has the canonical machinery to RNAi processing, or if they have it, is it functional? If Sclerotinia abscissum is able to process double-strand RNA and then induce the gene knockdown or maybe knockout. and the RNAi can be used for fungal control in the future. We are discussing here some promising strategies to control different kind of fungi. And this kind of question can drive us or drove us to perform some experiments to understand the pathogen biology and the possible use in the future of this RNAi. So to start to answer this question, I will now describe some experiment that I was doing and have done to try to figure out the behavior of RNAi in Sclerotinia abscissum. So I started using sequencing or next generation sequencing by HiSeq and PacBio, and searching for these proteins related to the RNAi machinery. For example, Dsrna, Argonaut, RD, RP, and other ones. And afterwards, I checked if these proteins are being expressed, just to be sure that it's not just present on the genome, but it's somehow expressed. So I checked it before I start another experiments with Sclerotinia abscissum. So they were there, and then I start to go further. So after this beginning, I planned a strategy to verify the functionality of this RNAi machinery and everything in Sclerotinia abscissum. So I use a simple schema here to show you what I was thinking that time and the process. Much more simple schema than the figure before from Professor Cole, but I guess it's quite easy to understand what I was thinking about. So here we have this fungal cell. It's simple draw, and nucleus and DNA. What I have done was transform this fungal cell with this Dsrna protein expressing vector. So after this transformation process, we have the DNA, the Dsrna DNA inside the DNA of the fungus or fungus DNA being transcripting and then translating in Dsrna proteins, fluorescence protein. After lots of fluorescence proteins, we have a red fungal cell. I think the microscopy is a good thing a good tool to literally see the things. So I really like to use microscopy because we can literally see the things happening or not. Yeah. So after this first transformation with the fungus with Dsrna express protein vector, I got some mutants. These mutants are expressing the Dsrna protein here. Here's the merge image. So it's constitutively expressing the Dsrna. Then what I have done was transform these Dsrna mutants, so a second round of transformation with this vector that induced the hairpin of Dsrna. So after here, yeah. Now we have the Dsrna gene inside the DNA, the fungal DNA, and also the DS hairpin DNA inside. So after transcription, this hairpin goes to the cytoplasm, and then it's processed. It's a complex process. Of course, I will not go deeply in these details. And after processing, we have this interfering RNA blocking the Dsrna mRNA translation. So showing this in my fungal, as you can see here, we have lots of knockdown and knockout here in this fluorescence images. And also I perform RT-qPCR to check the number of transcripts, the Dsrna transcripts, of course, reducing in comparison to the Dsrna wild type. I mean, this wild type is the mutant that express constitutively the Dsrna protein. So after look to these results, we can see that the Clathrin composition has the functional machinery for RNAi, and it means that it's possible or would be possible in the future as a strategy to fungal control. So it's a proof of concept here. So then some another questions come. So is this strategy useful for endogenous genes, for example, and how to test the effects in the pathogenicity? So to answer the first question, I selected some different targets, and we decided to target genes whose products are proven related to the fungal control, I mean the chemical control. In this sense, we select the same target genes at fungicide targets, and the selected genes were the succinate dehydrogenase subunits genes, the SDHA and SDHB, because these SDH inhibitors are used for control for many species of Clathrin. Yeah. So then another question, how to test these effects in pathogenicity. So for that, I used an in vitro system that I have developed, which make possible test this pathogenicity in the controlled environment. Yeah, so now we have the system and how to test it. So putting both together, here you can see that these mutants shows significant reduction of this number of transcripts compared to the wild type, not the Dsrna wild type, but the real wild type here. And now we have the functional and here are the pathogenicity test showing that these mutants are reducing or null pathogenicity in the citrus flower. And in this sense, in this scenario, we know that the function, this RNAi system is functional, and we have now different potential targets. And the next step is how to deliver it to double-strand RNA and induce the RNA interference. So there is many different strategies here to deliver it. Professor Kogel described some of this before, but I will just focus in the Higgs and Sigs in this presentation because I tested it in the Clathrin system, and I use it for all experiments, the same strategy, targeting the Dsrna transcripts as a proof of concept. Yeah. So I spread this this double-strand RNA in the fungal cell, and we see here some similar behavior, or similar reduction of Dsrna protein in comparison to or similar to the first images before. Yeah. So this indicates that the Clathrin system can uptake the Dsrna, the Dsrna from the environment. It's quite different experiment that Professor Kogel has showed before, and I guess Professor Heilingen will explore better the uptaking process next. So, and another strategy or delivery strategy that I have testing in Clathrin system was by Higgs. So here the host needs to produce the double-strand RNA targeting the fungal transcripts. Yeah. So this strategy here requires plant transformation and lots of process, which takes more time and hard work. Again, as I said, the target is the Dsrna gene, and as we need to inoculate the fungus in the citrus flower, we use a plant that or a plant variety that have earlier flowering. As you can see in this image here, we can see the flower or the flowering inside the tube. So it's really early flowering. So this allow us to analyze the fungal science really early, then wait until the citrus tree grow and produce some flowers. For that, I have the collaboration of Dr. Raquel Boscariol, who has a large experience in citrus transformation. But due to the pandemic scenario that we are living now, this experiment now stopped, or we could not finish this experiment to analyze if the host can silencing the Dsrna protein inside the fungal cell. So we hope that until the finish of this semester, we can literally finish this experiment to get some results. So our experiments demonstrated this functionality of RNAi and some delivery possibilities. But we think that it's necessary some most specific target gene or genes. So this is our new problem. So how to find some target genes? Then we back to the genome sequence, and we decided to explore more this genome and use the genome comparison to find some new species or specific targets in addition to another test that we are doing now. For this specific work, we have the collaboration of Dr. Ricardo Baroncelli from University of Salamanca, who is amazing bioinformatician and is helping us to analyze this data. Yeah. So in conclusion, and looking at this whole scenario, we tested, or we are focused, of course, in Sclerotinia abscissum, who is the guilt for the post-bloom fruit drop in citrus, and we checked this RNAi functionality, and now the next steps would be make some new targets or search for some new targets to silence a specific gene of Sclerotinia abscissum. For that, we will use the genome, and then after select the gene, select a best strategy. For example, spray-induced gene silencing, or maybe host-induced gene silencing. And at the end, validate this strategy and its efficiency, of course, and try to validate and use it as commercially or commercially scale. Yeah. So I would like to thank your attention, and specifically Professor Elena Beraldi and Bruno from Università di Bologna, Ricardo Baroncelli, Universidad de Salamanca. A part of this work was done during my PhD and postdoc, so under the Marcos Machado advisor, and at that time, and Professor Raquel, Dr. Raquel Boscariol for these transformation steps, and also Professor João Paes here from Efficacy, who is helping me now to adapt the things here. And of course, the finding agency who helped a lot and still help, all the iPlanta community, and for giving me this opportunity, and also my uni and the INCT Citrus. So here's my contact, my email contact. Feel free to send any question or doubts, and I'm open for questions. Thank you.
Thanks a lot. Sorry, I had was very nice to hear some data on Colectotricum. I don't know very much this species, but I know that other species of Colectotricum are quite tricky. So it's nice to have some evidence that RNA interference can work, and I hope it will be of stimulation for other species, because it's a big pathogen, this one. So we have one question here from Francis Funk, who is asking, on the slide named alternative control RNAi, why the silencing is not homogeneous? Namely, some IFE have fluorescence and some other do not.
Thank you, Francis. We were thinking a lot about this kind of result, but we think that it's not all the hyphae or the transcripts that are blocked or silenced by this. I'm reading here the chat. So we think that it's possible because there's two things. One is the promoter. So it's different, the promoter from the RNAi construction and Dsrna express protein. And it's not all the transcripts that are silenced by the RNAi. And also, what's the next part? Silencing. Maybe the production of the Dsrna protein is higher than the hairpin production. So that's the idea. We think about that. So it's a good question. We spend a lot of time to think about.
Okay, there are other questions.
I've got a quick question.
Yeah. Okay.
This is Chris Lawrence.
Cristina Azevedo saying, have you tried to spray the plants and infecting them after?
Yeah, Cristina, no. We just performed experiments in vitro in media. We haven't tried spray the plants yet.
Elena, there is also a question from Chris. I know.
Yes, someone's telling here. I guess Christian Lawrence, I guess.
Do you hear me?
Hi. Can you hear me?
Yeah, go ahead, Chris.
Yeah.
Thank you, Bruno. Yes, I just had a quick question in reference to Francis's question. Is Calotoptricum, this species, is it multinucleate per cell? And did you do a purification step after you did the transformation? You might have some nuclei that are transformed with the hairpin and some construct and some nuclei that didn't. So that may help explain why you don't get 100% silencing across.
That's a good point. Perfect. Thank you, Lawrence. That's true. Some species of Sclerotinia has more than one nucleus, and this hypothesis, I didn't think about that now, just not remember. But yeah, this is one of our hypotheses, but we did not perform this experiment to just use single nucleus. But thank you. It's a good point. We have talked about this before, but now I forgot.
There is one last question. Can you rule out autofluorescence?
There was not autofluorescence in the Clathrin-Mediated system, just fluorescence from the expression of the Dsrna protein that I put inside.
Okay, I have one last question. Maybe you mentioned it. I don't know if I missed it, but what is the size of the double-strand RNA? No, the one that you spray. No, what is the size?
This that I sprayed was there was different size that I tested because we have different constructions, but this one was 285, I guess. I'm not sure exactly now, but more than 250, less than 300. So it's in between.
Okay, thank you very much. So
thank you so much.
If there are other questions, we go on, and it's the turn of Professor Belmonte, Mark. He's a professor in biological sciences at the University of Manitoba in Canada. And he has studied as an undergraduate in University of Calgary. And then he moved to Winnipeg, where he carried out the PhD in plant science. And then where he is now as a professor. And his laboratory is dealing with Sclerotinia and the use of RNAi technology on this pathogen. So just go on, Mark, please. Thank you.
Great, thanks, Elena, and thanks to the other organizers of the meeting. It's a really great opportunity to be able to connect with so many other researchers from around the globe by just sitting, in my case, in my dining room. So what I wanted to do today was talk a little bit about our work that we are doing investigating the use of RNA interference, both as a spray approach or SIGs, as well as a transgenic approach or HIGs, in the control of a really devastating fungal pathogen called Sclerotinia. Now, before I get started, I do want to acknowledge my collaborator, Steve Wired, on this project. We've been working on the control of both pests as well as pathogens for a number of years using RNAi. And it looks like Steve will also be talking at the next webinar next week in the control of insect pests using RNA interference. So I am located in Winnipeg, Canada, which is the geographic center of North America, but it is also where canola was originally developed. Now, canola is not only grown here in Western Canada, but it also is grown all across the world in India, China, Australia, as well as a number of other countries across Europe, where it's also more commonly known as oilseed rape. In fact, canola is now considered to be the third most grown oilseed crop all around the world. And it's no surprise that we're seeing such a huge increase in the amount of canola that's grown. After all, it does have so many heart-healthy attributes. And so some recent data has shown that canola and the acreage of canola has actually increased over 20% globally over the past 10 years. But because canola is so profitable to growers, and growers are growing canola time and time again within the field, what that's doing is it's creating the perfect environment for various pathogens. And one of the most devastating fungal pathogens that affects canola, as well as over 500 other economically important crop species, is Sclerotinia. And a recent global survey shows that over 80% of all of the canola fields found around the world have some sort of sign of Sclerotinia infection. So as biologists, we really try to understand how the interaction occurs between the fungus as well as the plant. And so here what we have is the life cycle of Sclerotinia. What happens early on in the growing season is that we have these tiny little pebble-like structures called sclerotia. These sclerotia will then start to germinate and they'll release spores. The spores will then land onto the surface of the petals. These petals will then act as a food source for the fungus. The hyphae will then germinate on the surface of the petals. Those petals will then land onto the surface of the leaf. The hyphae will then start to colonize the leaf. It will then enter the vasculature of the plant where it becomes systemic. After the fungus then starts to colonize the stem, it creates architectural weakening of the plant. The plant falls down or it lodges, and we see a tremendous impact on overall yield for the grower. Unfortunately, there's no real true immunity against Sclerotinia, unlike some other economically important pathogens like blackleg, for example. We also have seen not great control using some of the more traditional broad spectrum fungicides. And these tiny little structures down here at the bottom called sclerotia can also be viable within the soil for over a decade. So there's an immediate need to find new, sustainable, as well as green technologies that we can provide and offer to canola growers all around the world. So one of the things that Steve Wired and I started to do is started to investigate whether we could use our knowledge of RNA interference in order to find new ways to control the fungus. So I don't really need to go through what RNA interference is, but quite simply is that we're interfering with the progression of the genetic information, specifically at the mRNA level. So what we do is we go ahead and synthesize a double-stranded RNA within the lab. That double-stranded RNA is then going to come into contact with the fungal cell. Once it comes into contact with the fungal cell, it's then going to enter the cell or be taken up through what we consider to be a mechanism known as Clathrin-Mediated Endocytosis, and I'll be providing some evidence into that mechanism, at least for long double-stranded RNAs. The double-stranded RNA will then enter the cell. It will be then recognized by Dsrna as well as all of the other RNAi machinery, and then it will then be broken down into a small RNA before it targets the mRNA of choice. So one of the first things that we did in order to identify new targets was to sequence a number of canola Sclerotinia plants, or canola plants that were infected with Sclerotinia. We then used and developed what's called a target identification pipeline that utilizes our knowledge of RNA sequencing analysis in order to take advantage of a number of different factors, including how high the gene might be expressed, how transcriptionally controlled it might be, so whether that gene is induced, that can help better identify and narrow down the list of targets that might be successful, both in the lab as well as in the field setting. Once we've identified these target sequences, we then use a computational approach to make sure that there is no off-target effects. We'll then synthesize our dsRNAs within the lab using an in vitro transcription method. The next step is to confirm that these dsRNAs do in fact target the mRNA of choice. We can then validate that using quantitative PCR. If we do see in vitro knockdown of the culture, we can then go on to the spray test. So we'll do both a spray induced on Arabidopsis as well as on canola. And then if that's looking good, then we can go into our transgenic approach. So one of the first questions that we had was, well, how long does it take for these molecules to work? So in this case, we treated Sclerotinia culture with 500 nanograms of double-stranded RNA. We then quantified the target mRNA knockdown in various targets. In this case, we're looking at 3 different targets. So one that's targeting the mitochondria, another dsRNA that might be targeting toxin biosynthesis, and then a third target that would be targeting protein modification. Each one of those mRNAs is then color-coded in either the gray bar, the purple bar, or the blue bar. And what we can see here is that after 24 hours, we can see a significant as well as profound reduction in target gene mRNA levels, and that this is maintained over 48, 72, as well as 96 hours. So what we could see here, at least from some of our preliminary data, is that we can see target knockdown within 24 hours or just a day. So how do we go about infecting our canola plants using dsRNA technology? Well, again, what we're doing is we're going back to what we would normally see in the field. So we're recapitulating field conditions in the lab where we are using the petals as well as the leaves in order to stimulate that type of interaction. So what we would do is we would go ahead and infect the petals with ascospores. We would then spray double-stranded RNAs onto the surface of the leaf. We would then allow for that petal and leaf interaction to take place, and then we would quantify the extent of disease that's found on the surface of the leaf, not only by looking at the phenotype, so how big the lesion is found on the plant, but also by quantifying the extent of target gene knockdown. So what this graph does is it shows a number of different dsRNAs that we screened through in the lab. In total, we have screened over 100 different Sclerotinia genes or Sclerotinia targets. Each one of these blue bars represents a different target. And in this case, we're looking at the lesion size compared to a water control. We've also looked at a GFP as well as a GUS dsRNA control, and we find that there, at least in our hands, there aren't really any differences depending on the type of control that you're using. So what we could see from this graph is that the vast majority of our targets that we identified do show some levels of RNA knockdown. But however, some of them actually show an increase, suggesting that these molecules might actually be negative regulators of the plant pathogen interaction. So what exactly does some of this protection look like? So again, Sclerotinia is considered to be a necrotrophic fungus. So it acts very, very quickly, and it produces these really large brown lesions on the surface of the leaf. So here we have an example of our control on the top left-hand side, where within a matter of days, we can see that almost the entire leaf has been consumed by the fungus. However, if we spray double-stranded RNA into this little area here on the leaf, depending on the dsRNA that we applied, either one that targets toxin biosynthesis, the mitochondria, or protein, they show varying levels of success. However, we do see, again, dramatic reduction in the lesion spread over a matter of a few days. So one of the other really nice things about using dsRNA or RNAi technology is that we can not only synthesize a number of molecules in a relatively short amount of time, but we can also extend and translate that knowledge into closely related fungi. So Sclerotinia is considered to be a white mold, and one of its close evolutionary neighbors is called botrytis, and botrytis is called a gray mold. So anytime you may have maybe left out some fruits or vegetables on the surface in your kitchen on the counter, and then all of a sudden you get all of these little gray hyphae that appear. So that usually is botrytis. So I know a number of us have seen botrytis at home, but in this case, we're looking at the control of botrytis in the lab on the Canola pathosystem. And what we did is we went ahead and we synthesized double-stranded RNAs of the homologs that we identified in Sclerotinia. We then allowed for that interaction to take place. And so each one of these bars represents a different botrytis target that was also successful in Sclerotinia. And what we could see here is that regardless of the target, so whether it's involved in detoxification, the mitochondria, or protein modification, we see a significant as well as profound reduction in the overall size of the lesion compared to, in this case, a water control. But again, we've done similar controls using a GFP or a GUS dsRNA. We then went ahead and quantified the amount of fungus that was found within the leaf, in this case, using quantitative PCR. And again, what we could see here is that when we apply a spray of the dsRNA onto the surface of the leaf, we can see again a dramatic reduction in the disease severity, and that disease severity correlates really nicely to not only the amount of fungus, but also the target gene knockdown within the leaf. So a lot of this data was published a few years ago by some of my graduate as well as undergraduate students in the lab, and of course in collaboration with Steve Wired. So we can now show that dsRNAs can in fact protect canola against two different closely related fungal pathogens, so Sclerotinia as well as botrytis. The next thing that we wanted to do was to take a host-induced gene silencing approach. So in this case, what we did is that we transformed the model plant Arabidopsis so that it would be constitutively expressing these hairpin RNAs. So again, if the dsRNA was successful as a spray, we then used similar targets. We then transformed Arabidopsis. And then over multiple generations, we tested how Arabidopsis was able to protect itself against Sclerotinia. So in this case, what we could see here is each one of these bars represents an independently transformed line that is overexpressing the hairpin RNAs that target mitochondria, toxin biosynthesis, protein modification, or some sort of pathogenicity factor, again, specifically within Sclerotinia. And what we could see here is the lesion size, where we can see anywhere between a 40 as well as a 75 or 74% reduction in disease severity. So this was really encouraging that not only the targets that we were able to identify using our target identification pipeline were successful as a spray, but now we can see that they're also successful using a transgenic approach in Arabidopsis. Now, in this case, we don't see any other phenotypic responses to the overexpression of these RNAs. And we're currently taking both a mRNA as well as a small RNA sequencing approach to identify how both the plant as well as the pathogen are interacting early on during the infection process. And we've also extended that to the transformation of canola, but I'm not going to show those results just for the interest of time. So we then functionally characterize these plants a little bit more. So what we're doing is we're comparing our wild type plants to one of our best performers labeled here as SS2. We're looking at both the lesion size or disease severity, fungal load, as well as target gene knockdown. And what we could see here is that not only do we see a dramatic reduction in the size of the lesion, so the amount of disease that's found on the plant, but we also see a corresponding reduction in fungal load, so the amount of fungus that's found within the leaf, as well as that the dsRNAs are in fact working because they are knocking down the mRNA that we had selected based off of our target identification pipeline. So what's nice is that not only are we able to see that these dsRNAs function as a spray, but now that these are also functional, the implanta taking a transgenic approach, meaning that these plants are durable not only across multiple generations, but they also offer protection across the entire life cycle of the plant, which in areas of the world where transgenics are more acceptable creates a lot more opportunity for the grower. So the next thing that I wanted to get into for today's talk was some recent data that we published last year, trying to tease apart the mechanism for how dsRNAs get into the fungal hyphae. So in this case, we're specifically focusing on Sclerotinia. And one of the reasons why we're interested in uptake is, of course, to try and get around any sort of potential resistance that might build within the fungus. If we can better understand how the fungus takes up these double-stranded RNAs, then not only can we figure out improved formulation, but we could also start to better identify some of the modes of action of these molecules. And again, so that we can prevent any sort of mechanism that might evolve over time where these fungi might develop resistance against these double-stranded RNAs in these different forms. So what we did is we created fluorescently labeled dsRNAs. So again, very similar to what some of the other speakers this afternoon, or in my case this morning, have talked about. So in this case, we're labeling a 250 base pair sequence, very similar to the sequences that we used in the previous experiments. And then we started to trace how the fungi was coming into contact with these fluorescently labeled molecules over time. So what we're looking at here is both the bright field as well as fluorescent micrographs 2, 4, 6, and 8 hours post-infection. So what I wanted to draw your attention to were these tiny little green dots. So these tiny little green dots, this is the edge of the fungal hyphae. And what we could see here is that the signal or the fluorescence is really starting to amplify and really brighten up within the leading tip of actively growing fungi. So not only do we see quite active levels of the dsRNAs directly at the hyphal tip, but we can also see increased activity or presence of these dsRNAs within branch points. One of the other things that I'd also like to talk about, and just to reiterate kind of some of the things that we've heard previously, is that this interaction isn't homogeneous, or at least isn't homogeneous in our hands using the Sclerotinia pathosystem. And of course, it's something that we're interested in better understanding why we're not seeing homogeneous uptake across the surface of the hyphae. So we then started to use some of our knowledge of how dsRNAs are being taken up within insect models, but then translating that into Sclerotinia. So we've known for some time now that Clathrin-mediated endocytosis is involved in dsRNA uptake within insects, but we had yet to provide any concrete evidence that it was also being involved in Sclerotinia. So what we did is we've taken both a fluorescent microscopy approach as well as a genetic approach to try and better understand and tease apart the possible uptake mechanisms of these long double-stranded RNAs. So what we first started to do was use various inhibitors of Clathrin-mediated endocytosis. In this case, we're using what's called chlorpromazine, or CPZ. Chlorpromazine is a pharmacological inhibitor of Clathrin-mediated endocytosis. So when we co-culture the Sclerotinia cells with this inhibitor, in addition to the fluorescently tagged dsRNA, we can see that the dsRNAs are actually stuck towards the outside of the cell, and that they fail to get into the fungal hyphae, so to get into the cytoplasm of the cell. We then went ahead and confirmed this using qPCR. So here what we have are two different dsRNA targets, one that targets thioreducin reductase and another one that targets an mRNA involved in the mitochondria. The gray bar represents the control. The blue bar represents our target. So our target is in fact knocked down for each one of the thioreducin reductases as well as the mitochondria. But when we co-incubate the Clathrin-mediated endocytosis inhibitor, CPZ, with our fluorescent marker, what we could see here is that there's actually no target knockdown. So we've been able to show this at least at the level of the mRNA. We then developed and synthesized a Sclerotinia that was overexpressing GFP. So what we could see here is that this GFP is found within the hyphae of Sclerotinia in culture. And then we synthesized a dsRNA that targeted the GFP. So the idea here was that if this dsRNA that targets GFP gets into the cell, we should then see a reduction in the fluorescent signal. And that's in fact what we do see here within this middle panel. And then we wanted to show whether Clathrin-mediated endocytosis was also involved in the process. So we co-incubated our GFP tagged Sclerotinia with CPZ, so that Clathrin-mediated endocytosis inhibitor. And what we could see here is that there's really no change in the fluorescent signal. So we took a very similar approach to different steps of Clathrin-mediated endocytosis. So whether it was involved in the formation of the coated vesicles, the blebbing off of that vesicle into the cytoplasm, or the unloading of the cargo into its various cellular compartments, at least at the genetic as well as taking an RNAi approach, or at the pharmacological approach using various physiological inhibitors and cellular inhibitors, we can start to dissect that Clathrin-mediated endocytosis is, at least in some part, involved in getting these long double-stranded RNAs into the fungal cell. But as we've heard before, there's really no receptor that's been identified to date, at least for these long double-stranded RNAs, and it's something that we're actively investigating. And we're also interested in better understanding how the long double-stranded RNAs, as well as some of the alternative secondary double-stranded RNA structures that Steve and I are working on, are processed within the cell at the cellular as well as the genetic level. So what I wanted to do today is talk a little bit about how we're driving RNAi innovation through discovery science. We know that Sclerotinia causes billions of dollars in crop loss to growers all around the world. So not only those that are growing oil seeds like canola as well as soybean, but also a number of other crops. We know that RNAi can specifically knock down its target gene, not only in vitro, but also as a foliar, as well as a transgenic application, and that the development of these RNAi technologies can help protect some of the world's most important food systems. So this was a large collaborative effort from not only my lab, but also Steve's lab. Here we have some of those students who were working on the project in these beautiful golden fields of canola that many of you have already seen. And of course, this work would not have been supported without our generous funders. Thank you very much.
Thanks, Mark. Really very nice talk. And yeah, there are quite a number of questions. The first is from Paula. Did you check the degradation of double-strand RNA? Mainly for those that fail in repress the target mRNA?
For the ones that did not show a positive reduction in disease severity, we did not study whether those dsRNAs were being degraded quickly. So in that large graph, in that case, we were just going through a screen. And so if they didn't work, we weren't terribly concerned about why they weren't working, but we certainly have a much better understanding for how some of them can be more successful than others at this point.
So second question is from Sema. What's the concentration of double-strand RNA or the volume of spray used for foliar application?
So that really depends on the molecule. So in general, we'll start our screen with 200 nanograms per microliter, and then we would spray that onto the surface. But depending on the target, sometimes you need less, and then sometimes you need more. So we've also gone up to 500 nanograms per microliter, depending on the formulation, and again, depending on the target.
Okay, then there is Christina. Have you tried more than one target simultaneously in the transgenic approach in Arabidopsis?
Not in the transgenic approach, but we certainly have stacked some of these molecules using the foliar sprays. What's interesting about this approach is that we have tried dsRNA targets that are part of the same pathway. So for example, that might be involved in sugar metabolism, or similar pathways that might be involved in the mitochondria, so respiration, or that might be involved in protein synthesis or toxin. And what we're seeing, at least from the targets that we've seen so far, is that there's no rhyme or reason. So if you were to put the same pathway, you don't get an added effect. If you put different pathways, sometimes you get added effects. And what we're seeing is that if you already have a very successful molecule in terms of controlling the fungus, it's difficult to add on to that using other molecules. So we're looking at molecules that might show intermediate success, where if you stack intermediately successful molecules, we're seeing a much better additive response compared to putting together some of our best quality molecules together. So that was just, at least for the molecules that we're testing in our hands, it's been a struggle, put it that way, to test all of these different combinations.
Anna is asking, are you using mRNA degradation confirmation alongside the small RNA and mRNA sequencing?
So we have not looked at the degradation of these mRNAs so far using the sequencing approach, but what we have been able to show using our to be honest, we haven't done a lot of the small RNA sequencing analysis yet. So we have the data sets, but we haven't processed them. But in terms of the mRNA, we can, in fact, at the sequencing or RNA sequencing level, show that the target gene is knocked down.
I see. Manfred is asking how long is the double-strand RNA stable and active when applied to the canola leaf surface?
So we've done a number of experiments, in this case that haven't been published, where we show that the stability of the mRNA on the surface of the leaf using a northern blot approach is at least stable for over 2 weeks. But in terms of its functionality, we haven't gone all the way that far. Usually the leaves will eventually die off with Sclerotinia because it's such an aggressive pathogen that the idea behind Sclerotinia is that you're not killing the pathogen. So these aren't killing molecules. Instead, what we're doing is we're just slowing it down so that the plant can mount its own defense and so that it can stay up straight. So as long as the plant can stay up straight and produce seed, you're okay. But the molecules that we have tested are not, they don't result in pathogen death.
Okay, we have another question from Marco, who says constitutive expression could have any penalty of plant performance.
Yeah, so that was one of the original things that we looked at. And so in our case, in both Arabidopsis as well as canola, because we have taken transgenic canola all the way through to the 3rd generation, and there's no effects, which was quite surprising to us because we thought that the plant would be overloaded with all of these foreign molecules that it would want to try to get rid of. And so in that case, we have not seen any effects, at least on the overall yield or quality of the seed.
Last question is from Francis, who is asking on the slide name, how long does it take for RNA interference to work? Do you expect your conclusion was based on the genes that you selected, or it is a general rule? And second, I assume you always started your experiment with conidia, correct? Do you have any explanation for the fungal tips that show stronger uptake?
So to answer the first question, so we've looked at a number of different targets, and all of the targets that we have looked at so far, not only in Sclerotinia, but also closely related targets, because we're also studying off-target effects, is that the molecules, we generally see a reduction within 24 hours in vitro. And so in vitro, of course, the molecules are surrounding the cells, and so it's a lot easier to get in. On the surface of the leaf, we do see knockdown of the target within 24 hours, although there is a possibility that it might be a little bit shorter, might be a little bit more. And what we've seen with some of our other experiments in the lab is that we do see target knockdown even earlier. So at the beginning, we chose 24 hours as just a ease of experimentation and design, but we do know that it can go much quicker than that. Francis, when it comes to your second question, you always started your experiments with conidia. Yeah, so we started out with the sporulating structures. And then what we did find is that it was always the really young advancing tips where we see a lot of fluorescence, but there was also a lot of fluorescence at the branch points of the fungus. It wasn't, as I showed, it was not homogeneous across the entire surface of the fungus, at least in Sclerotinia. That might change depending on the fungus, and we're learning everything more and more every day. But we also noticed that the size of the cell, like a smaller, thinner hyphae, had more fluorescence, and then the older, thicker hyphae did not. So that's also something that might explain why the results, at least from a spray with the necrotrophic fungus that grows so quickly, might be more variable. So we do see a lot of variability when we test these molecules on the surface of the leaf. And that also might be because the cells are no longer taking it up. The older, larger cells are no longer taking up the dsRNA as quickly as some of the small ones. So, it's certainly giving us a lot of insight into why we're seeing some of the results when we see what we saw using the fluorescence microscopy.
Yeah, so we have 1 minute, so I am asking also something. Well, actually, I was curious about this toxin target that you see that is more, at least at the beginning in the phenotype, it looked more efficient. This is, I mean, Sclerotinia is a necrotroph, quite broad spectrum. So that is, I guess, a host non-specific toxin. And do you see also different stability in a better stability in when you target the toxin instead of other?
Yeah, so one of the things that we found is that genes like toxin that produce toxins, they are quite highly regulated, and which means that, because they're so essential for the fungus, that there's a number of different transcriptional regulation steps that the fungus really wants to upregulate this, right? So it really wants to turn it on. So what we found in our work is that genes that get turned on or that are induced as a result of pathogen-plant interaction, they're generally not good targets. So while the mRNA might in fact be reduced early, it seems to overexpress later. And so what we find is that as the RNAs start to disappear and as they're consumed by the fungus and they're processed, but then the fungus continues to grow, then what will happen with genes that are under quite broad transcriptional regulation, that all of a sudden it's going to overexpress and can actually cause, cause reverse. So it could actually make the interaction a lot worse if you target genes that are under that type of regulation. So
yeah, I was suspecting that.
Yeah, so really what you're looking at are, you want to find targets that are usually moderately expressed. So almost like a housekeeping gene would be good targets for those that are interested, or genes that are also show low expression, but that aren't induced very easily.
Okay. Okay, then I have another one, but then I will leave it for discussion, maybe the final one. Okay, so we go on. We are in perfect time with Eileen. Professor Jean is from Riverside. Okay, Eileen is very well known for her pioneering studies on cross-kingdom RNA interference, and yeah, I will shortly present also her curriculum, her story. So she had a PhD from the Shanghai University, and then she carried out a postdoctoral period at Norwich at the John Innes Center. And then she became full professor of University of Riverside in California. Then I think everybody knows her work on cross-kingdom RNAi and on botrytis. And today she will talk about spray-induced gene silencing for disease control is dependent on the efficiency of pathogen RNA uptake. So she will talk about this different specificity of fungal pathogen species, I guess. So Eileen, please.
Yeah, hello everybody. I first would like to thank Elena and Bruno for organizing such a nice webinar to introduce RNA-based antifungal strategies for plant protection. And
Eileen, can you just speak a little bit louder?
Oh, okay.
Thank you.
Yeah, so first I would like to thank Elena and Bruno for organizing such a nice webinar. And then today I would like to introduce some advances in our cross-kingdom RNAi, and then also I would mainly focus on the plant protection using the cross-kingdom RNAi and the environmental RNAi strategies. So the pathogen we are working on is botrytis cinerea. Mark already introduced this, which can cause gray mold disease on almost all the fruits, all the vegetables, and many flowers. So it can infect more than 1,000 different plant species, cause serious damage, billions of dollars damage every year. So a few years ago, my postdoc Anna Weiberg and graduate student Ming Wang, they discovered that when botrytis infect the plants, it can trigger immune responses like any other infection. But what they found was that botrytis can deliver small RNAs into the host cell and use host Argonaute-1 protein to silence host defense responsive genes, and then to achieve very aggressive infection. So this basically is a cross-kingdom RNAi and was adapted by those aggressive fungal pathogens as an advanced virulence mechanism. So we were very happy to see that Amy Burke's lab from Edinburgh, they also observed the same phenomenon in a nematode mammalian interaction system. So what he found was that nematode can also deliver small RNAs, the parasite, the gut worm basically, can also deliver small RNAs into the mammalian guts, and then they can be taken up by the epithelial cells of the gut, and then which can suppress host genes involved in inflammation and immunity. We found even the target pathways are conserved because in our previous study, we found MAP kinase pathways are targeted by fungal small RNAs. Here, they also found that mammalian MAP kinase pathway signaling pathways also got targeted by the parasite small RNA. And recently, we also happy to see that even animal fungal pathogens, they can also deliver small RNAs and hijack host RNAi machinery in mosquito by binding to mosquito Argonaute-1. So the mechanism, the molecular mechanism is also the same as our plant fungal interaction. So it looks like now this cross-kingdom RNAi seems quite broadly applied in or evolved in different interaction systems. So then our next question is a cross-kingdom RNAi bidirectional? So then my other postdoc, Chiang Chai, what he discovered is that plant can also deliver small RNAs into fungal pathogens using these extracellular vesicles. And those small RNAs from the host can also silence virulence-related genes in fungus, and then achieve to control the fungal infection. So this basically is a co-evolutionary arms race, and the cross-kingdom RNAi have been evolved in both the fungal side and the plant side, and that this is like the small RNA trafficking is bidirectional. So the vesicles, we discovered that vesicles play a very important role in cross-kingdom RNA trafficking, but there are different classes of extracellular vesicles present in mammalian systems, although they were not well studied in plants. But in mammalian systems, they can be divided into three large subgroups. One is the exosomes, one is the microvesicles, and the other one is the apoptotic blebs. So they are divided based on their specific protein markers and their biogenesis pathways. So the exosomes, they are derived from multivesicular bodies, MVBs, and then their major protein markers are tetraspanines. So what we found is that the plants also has tetraspanin-like proteins, and that they are associated with a class of plant extracellular vesicles, and that they can be secreted. And we found that this class of vesicles, we use the GFP labeled, and we can visualize those extracellular vesicles. And then we extracted these extracellular vesicles from the leaves, infected leaf in the apoplastic fluid. And that these extracellular vesicles, the 80 tetraspanin 8 labeled vesicles, can be literally taken up very efficiently within 2 hours by the fungal cells. And then we can detect the host small RNAs in those fungal cells. So this, I hope you can remember in mind that the vesicles basically can be taken up very efficiently by the fungal cells. But in this experiment, since when we extracted the vesicles from the apoplastic fluid, although we can visualize these tetraspanin labeled exosome-like vesicles, but when we extracted many different, like all the vesicles. So in that case, we are not 100% sure if the RNAs transferred by these vesicles are indeed these tetraspanin labeled vesicles. So based on the mammalian studies, we found that the best and the most definite answer to address this question is to use immune capture to isolate these protein marker specific vesicles. So in that case, we would like to use the same approach because this is the highest standard we can get to purify a specific class of extracellular vesicles. So since we are interested in these tetraspanin labeled vesicles, which is considered plant exosomes, we generated a native antibody against the tetraspanin 8 because the tagged tetraspanin, both of the N-terminal end and C-terminal end of this tetraspanin protein are inside the vesicles, which will not be able to use for immunocapture, immunoprecipitation. So we generated the native antibody, recognized the out vesicular domain, and this antibody looks like very specific to tetraspanin 8 because it's in the tetraspanin 8 mutant background. We can no longer detect any signal. So this is the isolated EV. So we use this antibody to pull down the exosomes, the tetraspanin 8 positive exosomes, and this would be the purest vesicles we can get experimentally, and then we can detect host small RNAs in these vesicles. So this is the definite experiment to support that the tetraspanin 8 labeled subclass of plant extracellular vesicles, which we consider them to be exosomes, are responsible for the host small RNA delivery, host small RNA trafficking. And this part of work, together with another major part, we solved the mystery why those small RNAs are selectively loaded into the vesicles and transported into the pathogens. So this work was recently accepted in Nature Plants, or be out end of this month or beginning of next month. So in summary, we like a few years ago, we wrote this review. We discovered that fungal pathogen can deliver small RNAs into the host, but we predicted that small RNAs should be also delivered like a traffic from the host to the pathogen. But during that time, we didn't have real experimental data to support that. It's just a hypothesis. So I put a question mark on this. And then I also predicted that this kind of small RNA delivery and trafficking is present in many interaction systems, including parasitic plants and the host interaction, even the symbiotic fungal species with their interaction host, and between the insects and the host, or nematodes and the host. I put all the question marker there in 2015. But then now in this study, we found that plants can deliver endogenous small RNAs into the pathogen. So this question marker can be removed. And then also Mike Extol's lab, they found the daughter, the parasitic plants, can also deliver small RNAs into the host and suppress host genes. So all these questions. So now, like Uta and Ronald's lab, they discovered that that there are like also, so the mycorrhizal people, they found that they can use this host-induced gene silencing to manipulate the gene expression in mycorrhizae, because mycorrhizae, it's very difficult to generate mutants. So they use this host-induced gene silencing approach to do that. So in that case, the small RNAs expressed in the host should be able to move to the fungal, the mycorrhizae to introduce silencing. And then, so all these question markers now has been proven to be true. This cross-kingdom RNAi is the cross-kingdom RNAi is present in almost all the interaction settings. So the recent review we just wrote, just published at end of 2019, we used a similar theme and brought up a whole new set of question markers. And we predicted that extracellular vesicles play a very important role in this RNA trafficking or any communications between the interacting organisms. So now more and more colleagues and friends told me that they observed extracellular vesicles in these interaction settings. And the Ronald's lab, Uta's lab in Cambridge and Oxford, they found that there are a group of extracellular vesicles observed in the interface between the host and the mycorrhizae fungi. So I hope in the near future, all these question markers can be also crossed out. So that's like an update on cross-kingdom RNAi. Now I would like to introduce our recent findings on the plant protection using RNA approach. So this work was mainly previously done by Ming Wang and Lulu, and now is carried on by Jonathan and Rachel. So one of the previous questions asked by one of the attendees, they were asking if the Higgs can be used to control multiple genes. Yes, we did that, and we published it in 2016. So we think if the host-induced gene silencing is working, the cross-kingdom RNAi is effective, then it should be possible that we can use this approach to spontaneously control multiple pathogens. We can even custom design those transgenic plants that can target different pathogens upon the need. For example, like in Brazil, you have a certain pathogen species, then we can design the transgenic plant or the double-strand RNA target those particular species. And then in UK or in California, we have different set of pathogens, then we can custom design these transgenic plants or double-strand RNAs to target those pathogens. So in order to prove the concept, I asked my graduate student, Ming Wang, to generate transgenic plants that express small RNAs that can target Botrytis DICER1 and DICER2 and the Verticillium DICER1 and DICER2, because Botrytis is a foliar pathogen. In fact, the leaves and flowers on the plants, but the Verticillium is a root pathogen, infect the roots and then cause wilt phenotype. So I think this can separate the different parts of the plant, but we can monitor the disease symptoms. So we designed 4 different RNA fragments target individually DICER1, DICER2, DICER3, Verticillium of Botrytis and the Verticillium. The reason we do that is to try to avoid any potential off-target effect. So that's why we don't use any conserved regions to avoid off-target effect. We use specific regions for specific dicer. So then these transgenic plants express 4 fragments target DICER1, DICER2 of Botrytis and Verticillium. And then after we got these transgenic plants, and these transgenic plants express small RNAs pretty well. And then when we challenge these transgenic plants with botrytis, they displayed very significant resistance or tolerance to botrytis infection. And furthermore, when we infect them with Verticillium dilate, these transgenic plants stand very well in the soil when the wild type plant already completely died almost. So this is just to show you, provide a good example to show you that it's possible that we can use cross-kingdom RNAi to spontaneously control multiple pathogens. But even though host-induced gene silencing is working, but many crop plants do not have the available genetic manipulation tools, so you won't be able to easily transform them. And if you can transform them, sometimes it takes a long time, take years to generate transgenic plants. Moreover, GMOs are still a concern to many customers. And in Europe, this GMO was still banned, right? So in that case, we should develop a faster non-GMO approach that are eco-friendly to control those fungal or mycete diseases. So environmental RNAi was observed in C. elegans for many years and some nematodes as well, but it doesn't work in vertebrates or mammals. And it's not clear if it works in plants or fungi. So a few years ago, my graduate student, Ming Wang, I asked him to try to see if fungal pathogens can take up RNAs. So what he did was just to, on a plate, put the fungal spores with fluorescent labeled RNA, and including double-strand RNA and the small RNA, and then he found that fungal cells can take up RNAs pretty efficiently. And then since we needed to make sure that those RNAs are taken into the fungal cells instead of stick on the surface, we did the microcroconucleus treatment, and those signals are still there. Since that was like a new observation, we want to make absolutely sure, just in case, to avoid the possibility that the microcroconucleus treatment not thorough if the RNA is embedded in the fungal cell wall. So that's why we also remove the fungal cell wall and then do the microcroconucleus treatment. What we found is that these small RNAs and the double-strand RNAs are still present in the fungal cells. So that makes us confident that fungal cells can indeed take in the double-strand RNA. So in that case, we can design the double-strand RNA target pathogen-specific genes, virulence-related genes. Since we discovered that those small RNA effectors delivered by the pathogens to the fungal cells, to the host cells to suppress host immunity, those small RNAs are dependent on botrytis dicer 1, dicer 2. So the first genes we targeted is botrytis dicer 1, dicer 2. And then we found that we spray the double-strand RNA or small RNAs that target botrytis dicer 1, dicer 2, it can provide protection or inhibition, protection of the plants and inhibit the botrytis cause the gray mold disease compared to the wild type or the water or YFP controls. So I was also very happy to see that the 6 is working not only in monocot, but also not only in dicot, but also in monocot. Kelly's lab showed that they spray double-strand RNA target P450 gene can suppress Fusarium disease in barley. And then Mark's lab also showed that they can use double-strand RNA to control Sclerotinia and botrytis in Brassica nepus. So I'm very happy that both of them just gave a talk, but these are their original study. I think this provide very strong evidence to show that the double-strand RNA spray is indeed working. in multiple different pathogens. But every time when I give the talk, many people ask me, how about my virus? How about my fungal pathogens? So like many people want to know if this is applied to many different fungi or just a specific class of fungi. At the beginning, when we first published those fungal RNA uptake and the double-strand RNA mediated plant protection, most of people didn't believe it until later on more and more publications showed up. But then after that, people kind of switch from one extreme for like a not believing of it to the other extreme. They think every fungi should take up RNAs. So then they found, oh, my fungus didn't take up RNA, so they think this won't work, or this has very low efficiency. So to answer all these questions, we tested quite a few different fungal species, those ones that we can get hold of. So we not only tested botrytis, Sclerotinia, but also the rice fungal pathogen Rhizoctonia, and then also Verticillium and the Aspergillus niger. So what we found is that most of the fungal pathogens we tested, especially those very aggressive fungal pathogens, they can take up RNA very efficiently. For botrytis, Sclerotinia, and the Rhizoctonia, they can take up RNAs within hours, within like 2 to 6 hours, and then after 10 hours. So these pictures are taken after 10 hours. So you can see a mass amount of RNA uptake. These pictures were taken after the micrococcus nucleus treatment. So to address one of the questions earlier, these RNAs are indeed get inside the fungal cells, because otherwise, if they are stick on the surface, they will be degraded by the micrococcus nucleus treatment. And then for Verticillium and the Aspergillus, they take up the RNA slightly slower than botrytis and the Sclerotinia and the Rhizoctonia. They can reach to this level after 10 hours, like around 10 hours. But at 2 hours, you only see very faint signal or hardly any signal. So then for those pathogens we tested, since they can take up the RNA very efficiently, we tested their efficiency, the plant protection efficiency, by using different double-strand RNAs. So in our recent 2018 science paper, we discovered that the host small RNAs can target a set of protein trafficking genes in fungi. So we now designed the double-strand RNAs targeting these 3 genes involved in fungal trafficking pathways. And we also use our like a classical dicer1, dicer2 double-strand RNAs. And we found that when we spray these double-strand RNAs on lettuce and the carrots, greens, those vegetables, we can like control the Sclerotinia infection pretty effectively compared to the YFP control. And then also the same set of double-strand RNA, but target the Aspergillus genes. So the sequence is different, although the genes are the same. We found that these pathways are also important for the virulence of Aspergillus niger, and the double-strand RNA targeted these genes can also confer protection for tomato, grape, and apple. So those fruits. So and in collaboration with my former postdoc Dongdong in China, they tested these double-strand RNAs that target these two trafficking pathway genes in Rhizoctonia solani, and then they found this double-strand RNA can also inhibit its infection. It's pretty obvious compared to the YFP double-strand RNA. So that means majority of the fungal pathogens can efficiently take up RNAs. So in that case, this double-strand RNA or small RNA spray mediated plant protection should work for most of them. But one challenge is that the double-strand RNAs have a limited plant protection because they are easily get degraded in the environment. So in our hand, we found the naked double-strand RNA can only last for probably 5-6 days. Sometimes we also see it can be effective after 10 days, but it totally depends on the environment. So I was very happy to hear that Mark can also see the naked double-strand RNA to be effective for up to 14 days, right? But no matter what, the naked RNAs are not very stable in the environment, couldn't provide long protection to the crops. So in that case, we should develop some strategy, approaches to protect those double-strand RNAs or siRNAs in the field, in the environment, or on the crops. So we took the approach to develop nanoparticles, including inorganic nanoparticles and organic nanoparticles to protect those plants. So we developed, since we show that extracellular vesicles can protect the small RNAs and which can also be efficiently taken up by the fungal cells. So we first developed those artificial nanovesicles, like the liposomes the mammalian people use for drug delivery. So we tested those DOTAP, glycerol, and then like the PEG different formula, and then we found use different lipids, different ratio with the double-strand RNA. We can see that the artificial vesicles can provide protection. Even we use micrococcal nucleus treatment, these RNA are still there, but the naked RNA will be degraded very quickly. So in that case, these double-strand RNAs can be protected by these nanovesicles. But since those ones are for medical use, so they are pretty expensive, but it's not practical for apply those formula to the agriculture uses. So in that case, we also try to reduce the cost to see if we can remove PEG, which is expensive, or change these lipids to some cheaper lipids. So then now we also figured out the good formulations that can make this protection and delivery work efficiently, and to extend the longevity of this double-strand RNA. And this is some examples to show you that this artificial vesicle itself won't do anything, but if we package the double-stranded RNA in these artificial vesicles, they can confer protection up to 10 or like 2 weeks. So this picture is only 10 days because those detached fruits can only last for 10 days, but in grape leaves, and then they can last longer, like up to 2 to 3 weeks. And this is the quantification. We can show that the genes are silenced in the relative lesion size is significantly reduced. But in addition to those organic,
to interrupt you, we have to move on quickly. So
yeah, this is the last slide.
Yeah. Okay, perfect. Okay.
The nanobioclay, the inorganic particles can extend the longevity to 2 to 3 weeks. This work is in collaboration with Nina Mitter from Queensland. So then this will be the new generation of RNA-based fungicide, which can address these problems that most of the current used fungicide already generated the resistance or partial resistance. So this is a safer approach. Okay, so this is one thing I want to mention in the end, is that not all the fungi can take up RNA. The Clathrin, this is different strain from the previous talk. We found that the Clathrin cannot take up RNAs like at all. After we use the micrococoa nucleus treatment, the RNAs are completely gone. So the double-strand RNA won't confer protection. These are all diseased. So this is to show you that spray-induced gene silencing will depend on the efficiency of the 6 depends on the efficiency of the RNA uptake by the fungi. So in that case, if you want to see if the double-strand RNA will work on your particular fungal pathogens, you should check their RNA uptake efficiency. So this is the group of people I would like to thank, and these are all my collaborators and funding support. Thank you very much.
Thank you, Eileen. Very nice. Was interesting to see this Coleotricum reluctancy to uptake double-strand RNA, but Coleotricum is a very interesting genus, as a very, very much variety of species. So, okay, I don't see any question in the chat. Maybe it's a bit late too, but we can go on, and then let's see at the end during discussion. I'd like now to leave room to Luca Capriotti. He's last year PhD student in the Department of Food, Agriculture and Environmental Science in the Mark Polytechnic University. And he's working in the lab of Bruno Mezzetti, and he's going to present some part of his PhD work that was also spent for some time in the laboratory of Eileen. So the title of the talk is Higgs and SIG's Approach to Induce Resistance Against Botrytis cinerea in Strawberry and in Grapevine. Thank you, Luca.
Thank you to everybody, and good afternoon to everybody. Thank you for the possibility that you give me to present today.
Can you make a full screen?
Yes.
Thanks.
Grain mold disease caused by the ascomycete Botrytis cinerea has no apparent host specificity, as mentioned by previous speaker, and can infect more than 1,000 plant species. The pathogen is found worldwide and can cause the disease in many fruit, flower, and leafy vegetable crops. Botrytis cinerea is classified as a necrotroph, in which they prefer to infect damaging or senescing tissues. The life cycle of Botrytis cinerea starts mainly in the form of sclerotia after overwintering in the soil or in dying organs. The fungus require a temperate and damp condition for conidia release, germination, growth, and establishment of infection. This opportunistic fungus can actively or passively penetrate host tissue, and infected fruits and succulent stems became soft, watery, and light brown. Furthermore, botrytis might also be so successful because it can combine apoptotic to necrotic strategies. Gray mold disease on strawberry represents one of the most devastating diseases, treating the production of strawberry, especially in the greenhouses. Pre-harvest losses ranging from 50% under normal condition to 90% under severe condition. The control of the aforementioned disease is generally achieved by the application of chemical fungicide. Fungicides in Italy are the most common plant protection product used in agriculture. And more than 50% of all these plant protection products are considered fungicide. Of the whole fungicide, more than 60% are used in vineyard. The economic cost and also the agroecological and environmental negative impact related to such application led to a search for alternative strategies, such as the manipulation of host defense mechanisms. An alternative approach to the time-consuming conventional breeding is the application of post-transcriptional gene silencing. RNAi is an endogenous cellular process which has the main function in gene expression, inhibition, or suppression, activated by the presence of double-strand RNA molecules. Long double-strand RNA molecules can be recognized by Dsrna protein that cleave it into small interfering RNA. These small interfering RNA bind with the argonate protein, the part of a silencing complex that separates them into two strands. The passenger strand is degraded, that the guide strand serves as a search probe to link risk complex with complementary RNA target. During the coevolutionary arm race between plant and fungi, successful fungi are able to send small interfering RNA as effector molecules that are able to silence host immunity genes. Also, plants can send small interfering RNA, especially through the formation of extracellular vesicles that are absorbed by fungal cells, where they knock down virulence gene expression. Candidate genes capable to limit pathogen aggression or improve plant defense strategies can be observed during the pathogenesis with the fungal causal agents. RNAi-based gene strategies can be exploited by improving plant resistance or through an introgression of an hairpin-based gene construct in the plant genome, or through the exogenous application of double-strand RNAs. In the host-induced gene silencing, as a consequence of a transcription of RNAi-based gene construct, a long double-strand RNA is formed that is recognized by Dsrna protein that cleave it into small interfering RNA, which knock down related gene expression. In spray-induced gene silencing, the external application of double-strand RNA could lead to the absorption of these molecules, and depending on the delivery method used, these molecules can be processed by both RNAi machinery of plant or fungi. The most important and known delivery method used for SIGS approach is petal absorption, trunk injection, low pressure spray, and high pressure spray. Eileen Jean and collaborators discovered that Dsrna is able to send small interfering RNA into host plant cells and utilize host RNA machinery to silence host immunity genes. They show that expressing small interfering RNA that target Dsrna gene 1 and 2 of Dsrna can efficiently silence this kind of genes. And the application of these double-strand RNA molecules on the surface of detached fruits, vegetables, and flowers led to a reduction in pathogenicity and growth. They also proved that Dsrna are able to uptake double-strand RNA that are present in the environment, as you can observe here, where they performed an experiment in which they labeled double-strand RNA with a fluorescence. And after a few hours, Dsrna, Hyphae, and Conidia showed a great emission of fluorescence. Through host-induced gene silencing, it is possible to inhibit fungal invasion and limit virulence gene expression through host-derived small RNAs. These small RNAs are constitutively expressed on the transgenic plants. Using a gene construct that was designed by Wong and collaborators in 2016, we did genetic transformation trials using PL-scale plasmid and GV3101 Agrobacterium tumefaciens strain. Agrobacterium-mediated transformation was performed on 2 cultivars of strawberry, Sveva and Romina, and 1 cultivar of grapevine, Thompson seedless. For both grapevine and strawberry, we use a gene construct that targets Dsrna gene 1 and 2 of Dsrna. Then, in strawberry, we use in addition another gene construct that targets both Dsrna gene 1 and 2 of Dsrna and Verticillium dahlia. To obtain high performances in genetic transformation efficiency, it is essential to follow very efficient regeneration protocol that were based on de novo shoot organogenesis. This organogenesis is a process that consists in the production of unipolar structure starting from already differentiated material. For strawberry, we followed a regeneration protocol described by Cappelletti and co-authors in 2015, in which de novo shoot organogenesis was induced by the performance of some cuts in the adducted surface of strawberry leaves that were grown in vitro, going to callus formation. For grapevine, we used a regeneration protocol that was described for the first time by Mezzetti and co-authors in 2002 and optimized by Sabatini and co-authors in 2019. This meristematic vegetative structure was a structure full of meristematic bud. We obtain some slices, and briefly, slices of meristematic bulk and leaves of strawberry have been placed in contact with Agrobacterium inoculum for 15 minutes, followed by culture and selection in selective media. This procedure may require several months because we genetically transform fruit crops. But at the end of this procedure, we obtain putative transgenic lines that are resistant to cannabis and enriched media. In grapevine, we obtained several green calluses that were able to proliferate and grow on selective media. From these calluses, we obtain elongated shoots that were stimulated to root in vitro. And once have been rooted in vitro, these shoots have been acclimatized to the greenhouses. A molecular characterization has been conducted in 4 putative transgenic lines of cultivar Thompson seedless, and we amplify the sequence of the promoter 35S that amplify at 340 base pair. The molecular characterization of these lines allow us to identify 4 independent transgenic lines, and genetic transformation efficiency was equal to 0.67%. Inoculation tests are needed to validate the resistance, the increased resistance against gray mold. A similar procedure has been followed for strawberry, in which calluses have been developed in shoots, and these shoots were transplanted in greenhouses. From this experiment, we observed that Sveva had a regeneration potential higher than Romina. The genetic transformation efficiency was reported as the number of putative transgenic lines that regenerated by the total number of explants treated, and the molecular characterization have been conducted on 6 lines by each gene construct used of cultivar Sveva, and amplifying the 35S sequences. Molecular characterization of other putative transgenic lines, especially for Romina, and for amplifying the hairpin-based gene construct are now in progress. 6 have been documented to trigger RNAi in planta, and long double-strand RNA or small interfering RNA can be sprayed on the surface of the plant and on the fungi, and can carry and trigger RNAi in planta. The aim of the experiment that we conducted now was to determine the efficacy of double-strand RNA-based treatment when it was applied on the surface of grapevine leaves that were grown in the greenhouses. This treatment have been conducted 1, 7, 14, and 21 days before challenge the plants with the Botrytis inoculum. After this data set, the grapevine plants, cultivar Cabernet Sauvignon, have been subjected to infection in inoculating 1 per 10 to the 5th conidia ml. 4 days post-inoculation, we did the image acquisition where we analyzed the necrotic lesion size that have been recorded in the detached leaves, and the relative biomass quantification through quantitative real-time PCR. 6, and the topical application of double-strand RNA emerging as an appealing alternative to genetically modified crops. However, the major limitation of this technique is the short window of protection post-spray. We observed that when we applied the double-strand RNA naked treatment 1 day and 7 days before Botrytis inoculation, we observed a strong protection in detached leaves, as we can observe also by the necrotic lesion size measurement and the quantification of the biomass of Botrytis in area. Carrying out this treatment 14 days and 21 days before inoculation, it is still possible to observe a strong protection that was provided by double-strand RNA naked treatment, as you can observe by the necrotic lesion size measurement, in which leaves that were treated with the double-strand RNA showed a significant lower necrotic lesion in the surface of the touch leaves. This experiment has been replicated 2 times, inoculating 5 leaves for each treatment. A different discussion must be made for fruit. In this case, grapefruit that were acquired on the local supermarket have been treated using in vitro synthesized double-strand RNA, 20 nanogram microliters at a concentration. And this treatment was performed, and 7 days after, we did Botrytis cinerea inoculation by a drop treatment. 4 days post-inoculation, we did the observation. We observed that double-strand RNA treatment can efficiently protect the surface of the grapes, the grape berries, when it was applied 1 and 3 days before inoculation. But when it was applied on 7 days, this efficacy started to be lower. Also in strawberry, we performed a similar experiment that was replicated 3 times. In strawberry are generally more susceptible to fungal attacks due to their poor firmness. And in this case also, we observe similar results. Double-strand RNA-based treatment seems to be effective when it was applied 1 and 3 days before inoculation, less when it was applied 7 days post-inoculation. Although we demonstrate that spray-induced silencing seems function to protect the touch leaves and fruits, few reports are now available in literature about the external application of double-strand RNA in planta. And for this reason, we used strawberry plants that were grown in pots in the greenhouse environment. And these plants have been subjected to 5 different treatments. The negative control, it was the water, 3 double-strand RNA-based treatment at different concentration, 10, 20, and 50 nanogram microliter, and the positive control that was the fungicide. 1 day after, we perform as a low-pressure spray treatment the botrytis infection, applying 1% to the 6 conidia ml. The disease progression have been monitored at 4, 10, and 14 days post-inoculation. This experiment has been replicated 3 times, treating 5 plants for each treatment. At each data observation, the disease severity have been calculated, assigning at each fruit that was attached to the plants a disease severity index. There were 5 disease severity indexes that were based on the affected area of the fruit to the total area of the fruit. In this case, we only evaluate the disease severity on the fruit because on the leaves we observed only sporadic necrotic lesion size. We also saw and calculate the parameter of the disease control value. That was a comparison of the disease severity recorded after a specific treatment to the disease severity that we recorded in the negative control. Our results suggest that double-strand RNA-based treatment, especially when it was applied at the highest concentration, in this case 50 nanogram microliter, can protect plants and reduce the presence of Botrytis cinerea, especially at 4 dpi, when we reached a control value about 60%. This efficacy starts to decrease at 10 and 14 dpi, as we observed after the application of the commercial fungicide. Among the other double-strand RNA-based treatment, we recorded a strong variability, maybe due to the RNA's activity that limit the availability of double-strand RNA that can be taken up by the fungal cell. Our data suggests that the application of double-strand RNA, especially at 4 dpi, can have an effect very similar and comparable to those obtained after the application of a chemical fungicide that was ordinarily applied for the control of the gray mold in the greenhouse environment. For concluding, in host-induced gene silencing, there is the constitutive expression of double-strand RNAs, and on spray-induced gene silencing, these molecules are applied externally to the crops and to the pathogens. 6 is a GMO-free strategies and that allow to overcome the regeneration recalcitrants that are typical for some genotypes. Some genotypes are not able to regenerate very well. And in 6 approach can be applicable in few months. Conversely, this approach requires a long time, especially for the constitution of a transgenic line, may require from 1 to 2 years, especially for woody plant species as grapevine, and require efficient regeneration protocol that are not always available for all the species and all the genotype of a species. And also transgenic plant are now classified as GMO in European Union. Probably host-induced gene silencing line are probably stably resistant to the pathogen. Then in spray-induced gene silencing may require multiple application of double-strand RNA molecules for prolonging the window of protection to the crops. Regarding the host-induced gene silencing approach, our aim is to obtain more transgenic line of strawberry and grapevine and to extend this genetic transformation to other genotypes. It is essential also to evaluate the protection effect against gray mold, especially testing the touch leaves and fruit of transgenic plants. Regarding the spray-induced gene silencing approach, it is fundamental to stabilize double-strand RNA in a formulation in order to prolong and extend the window of protection, and also to evaluate the probable use of the externally applied double-strand RNA molecules as valid tools for ordinary pest pathogen management, for example, through the application of these molecules on the greenhouse environment and on the open field environment. Thank you for your attention.
Thank you, Luca. Very nice. We had an overview of how this molecule can work really in reality, no, with the plants, okay, in the greenhouse, but yeah, in pots, but it's really, how do you say, eloquent for future application in field. So thank you very much. Are there questions for Luca? No, so I am sorry I didn't see before a question for Eileen. Okay, there is one question for Luca, so let's go for this one and then we go back to the question for Eileen that I didn't notice before, I'm sorry. So this is coming from Marco and he's asking, could the RNAi interference SIGs techniques be used for the control of botrytis in strawberry post-harvest?
Thank you for the question. We validate the use of double-strand RNA treatment in post-harvest in grapes and strawberry. And we recorded that the application of these molecules must be very intense because we saw that after 3 days, the efficacy of these treatments start to decrease. And when was applied 7 days before the inoculation, we did not observe any difference with the control treatment. Then, yes, there is a slight, I think, protection that can be provided by these molecules, but not longer than 3 days. In my case, in these cases.
I might add something to this because we have to keep in mind that most of the infection, the majority of the infection that we see in post-harvest on fruits comes from the field at the flowering stage, because Botrytis is capable of a very long quiescence on grapevine too. So it is possible that the application in field then can have good response also for for the subsequent stage at post-harvest phase. So yeah, so let's go back to the question for Eileen. It comes from Francis. Can you please talk about the current progress on understanding the secondary amplification of double-strand RNA in fungi?
So in fungi, there are RNA-dependent, like the RDRPs, RNA-dependent RNA polymerases, but we didn't follow that up very closely because my former postdoc, Anna Weiberg, now he is individual PI in Munich. He is following up on the Botrytis, Argonaut, and the RDRPs. So I think he probably have some results to answer your question. But I believe if they have RDRPs, there should be some amplification steps. But whether if this amplification is also triggered by 22-mer sRNAs, we don't really know. So this needed to be investigated more precisely.
Okay, thanks, Eileen. So is there other question or issues, comments that you want to bring to the discussion? It's late, I know, so, but few minutes.
So I have a question for Kelly. Kelly, you said you observed RNA uptake into the fungal cells. So you use the petrol from the to place the leaves on the RNA solution. You see the uptake, but did you see if on the surface of the leaves any uptake to the epidermal cells or mesophyll cells to the level that you can visualize them?
So we see the fluorescence of the RNA on the leaves, of course, and we also see a penetration of small RNAs. Yes, if this is your question, and this is mostly in the xylem. Yeah, associated with xylem, not associated with the phloem.
So in your xylem experiment, you basically cut the bottom of the petal of the leaves, and then you put that wounding site in the RNA solution, so the RNA got sucked up in the leaves. So that's perfectly making perfect sense. They are in the xylem, but I was wondering if you just spray the RNAs on the intact leaves, there's no wounding site. Do you see the efficient uptake of the RNAs into the fungal cells, like the epidermal cell layers or mesophyll cells?
You mean in the fungal cells? Yeah.
Oh, sorry, the plant cells. No.
In the plant cells or in the fungal cells?
Plant cells.
In plant cells.
Fungal cells we see.
Yeah, I showed you some pictures that were taken from reporter plants where you have a phloem reporter, the SAC2 promoter, yeah. And as far as we can see, there is no overlap with the phloem and the labeled RNA when taken up from the leaf surface. The RNA can only be detected in the xylem. But these are experiments with Arabidopsis. It might be different with with cereals or so, or with rice, maybe.
Thank you.
So maybe I can comment on that. We tried hard to get something into the cell, and with the siRNA it worked fine. And now the carbon dot method is really a method where you can get small RNAs into the the cell of a leaf by spraying, by low pressure spraying. All other methods so far failed.
So the carbon dot itself can get into the plant cell. So that make people wondering if they can cause any toxicity issues, like how much the plants can tolerate to have carbon dots inside the cell, right? Although they can deliver RNAs, but how can they be processed or metabolized, removed from the plant cell? Because you don't really want to have these carbon dots in the cell. So that's I think is a limitation for using that method.
Okay, this could be, of course, an issue. On the other hand, they are non-toxic, and I don't know the half-life of the carbon dots, but if you think about plants producing fruits, maybe, for example, grapevine, if you spray them in the time when you have a pathogen attack, it might not be in the fruits, for example, or in the corn. This has to be tested. But if you talk about the uptake of RNA into the plant cell, so far is high pressure spraying or carbon dot method that really works.
Mm-hmm. So the normal spray, you don't see any uptake?
No.
Yeah, that's similar to what we found. We found there are some uptake for sure, I think, because you can silence the endogenous genes. But the problem is the level is not high enough to the level that we can see the fluorescence RNA. So we did even the protoplast to make sure. So in the protoplast, we can see very faint signal. But that's why I was wondering if you guys can see it. But they certainly should get in, because Nina Mitter's lab, they saw this spray the double-strand RNA can silence virus, right? So the virus is inside the plant cell. So
you have to carefully read the paper. They always, when they did these experiments, they used the carborundum wounding, and by that you get it in. That's true, but not by simply spraying on the surface. Nobody has shown that this really works, and you can only show that if the double-stranded RNA is processed by Dsrna into small interfering RNAs, that they're indeed in the cell, and it would be immediately processed. And this, if you look at all the literature, this has not been demonstrated, only in a few papers, and they use the double-stranded RNA without anything, so no wounding or no other compounds as a negative control. In all the papers, this is the negative control. And we did really, also with the carbon dots, we were not able to get in double-stranded RNA, and not only in our hands, but also in the group that published this work. When the double-stranded RNA was longer than 50 nucleotide base pairs, they failed to get it in as an efficient silencer for a reporter gene. So we have to be really, really careful with saying it's in the cell before you don't demonstrate that it's processed into small interfering RNAs. I don't believe anything.
So then Kelly just said, without wounding, you can observe the RNA get into the xylem?
Yeah, we are talking about 21 nucleotide RNAs labeled, yeah, fluorescent labeled. And if you, we did a drop application in this case, and then you see it in the xylem. There is no doubt, because you see we have these reporter lines, and the question is how far it goes in the leaf, of course, yeah.
So you drop on the surface of the leaf?
Yeah, it's just on the surface, and there is no wounding in this case.
Yeah, but if you take a lower part of the leaf with as we did with the trunk injection, or with the endothelial absorption, then it goes to the xylem, but does not go out. And the point is that if you spray it on the upper side of the leaf, then it can enter, of course, the xylem system, because which cells are connected to the xylem. This is the Spaltöffnung Just tell me the
apoplast.
No. Gabi should just help me. The English word for Spaltöffnung.
Stomata.
The stomata, yeah. Then you can enter this in the stomata, and the stomata are directly connected to the xylem. When we did the trunk injection or petiole absorption, we could see that this the fluorescing or the labeled RNA was visible in the stomata cells. So, and this on the opposite way will also work. If you spray the stomata, you will get entry into the xylem system.
Yeah, then if you, this statement, you see, we agree completely. It is only in the xylem, yeah, and not in the, so far we don't detect it in the phloem. So therefore, yeah.
That's always the point, to get it into the phloem or then into the cells by surface application is not an easy thing to do.
Yeah, we agree to this.
Okay.
I have another question. If any other one has any other question, I have some question. Well, one question for Mark and one also for you, Kalle, on the fungal side this time. Was wondering about this localization that you see about the spray Dsrna at the tip of the fungal growth, and also at the ramification point, no? Am I right?
Yeah, that is more or less the same picture as in like in Sclerotinia.
Exactly. No, I was wondering if this was due to two different effects, because at the tip of the funga of the hyphae, we know that, I mean, that is the point of the very, very active growth. So a lot of vesicular traffic is going on there. And maybe also and also the cell wall is very thin and is very weak. So maybe there could be really a kind of effect due to these processes, whereas the ramification could be due to another effect, much more simple. Maybe I'm saying a very stupid thing, but could be just the steric effect, that they are less easily washed away. And then they just stuck there because it's like a corner. I don't know.
Yeah.
Yeah, the fluorescent molecules that were in the corners, they were inside the cell.
Okay, sorry. Okay.
So
They were looking at the surface. Okay. Mm-hmm.
When we did the Z stack, they were certainly inside and they tended to accumulate in regions. The molecules did not enter the vacuole.
Okay.
At least in our own experiments, but that might be different under different conditions. But as far as we saw, they were largely found within the cytoplasm, and then they did become more broad over time, but we were trying to initially see what was happening at the very beginning. Okay, thanks. Then I had another question for Kalle about you see that the Dsrna can be transmitted from the spores to the germ tube and also to the generated hyphae, no, of the fungi. But have you checked if this molecule, this RNA, can also go to the next generation spores? To the spores that are generated by those hyphae?
Yeah, that's a good question. No, we did not look at it. We did not wait for a longer time, but this is probably an interesting question. Yeah, we did not do that.
Okay, thank you. There is some question in the chat for Mark. do you think some of the bright fluorescence is located in septa?
Yes, definitely. So we also see a lot of accumulation of fluorescence around the septa that are found within the cells. That was something else that occurred that was very obvious from the very beginning in terms of the intensity of the signal. The reason why, I'm not sure, but it's another one of those things where, you know, if we could dissect the cellular movement of the molecules, and especially now that we have alternative structures that we're working with that do not seem to operate through Clathrin-mediated endocytosis, could be quite interesting. And I think Steve will talk more about that next week.
Okay. Other points? Well, if not, I think Bruno wants to say something about the picture.
Okay, yes. Well, first of all, if you have finished, I want to thank Elena for coordinating this, but also all speakers, because it was really interesting. For me, I'm not a pathologist. I'm trying to follow the discussion and see how we can transfer the knowledge you are developing in plants, in cultivated plants, like in particular fruit, like Luca was showing. So for us, it's really interesting to know all this knowledge and trying to understand how we can interact in developing this new knowledge in a more concrete way, because I think that the issue of controlling disease in the next period will be really important for agriculture in all over the world. So we need really to find new solutions. And I think this technology can be really a solution that can help to solve this problem. I don't want to enter on the debate about 6 and Higgs, so GMO or not GMO. I would say that as a scientist, we should keep all these both the two possibilities, and take the results and show what are the results, and then see what can be the best solution to be applied in order to help in solving this problem in agriculture. So that's my final comment related to this topic. Well, if there are no other comments, I would, as we are doing, everything is recorded. And before to stop to record, if people now still present want to turn on the camera, we can take a group picture.
Well, let's also to thank all the speakers. I forgot. Sorry, the was very interesting for me. Really very nice. Thank you.
Yeah, thank you again. Well, very top scientists, a lot of people really have really good discussion. Okay, now I see holes and nice faces here. Let me take the picture. And cheers. And thank you. I think that I got it. And yeah, I think that we can stop here. And please remember that we are organizing other seminars, webinars. The next webinars, it will be on insect, and actually Guy Maggie, that is here connected, and Olivier are organizing it for Tuesday, February 16. So remain tuned on the program of iPlanter webinars, and I hope to see you connected again next February 16, and maybe more other people that are specialists not on fungi but on pest control will be connected with us, and then continue with the other webinars. So let's stop here. It has been a long afternoon, and enjoy your day, enjoy your time, take care, and I really hope to see you again soon, at least virtually. Okay, bye-bye. Let's go.
Bye.
Bye. Bye. Thank you.
Bye-bye.
Thank you.
Tschüss.
Thank you all.
Thank you.